Welcome friends. Today we will start with the second unit, that is cell biology, and this is the first class of cell biology where we will discuss about membrane structure and function. So let's begin. So under this topic, membrane structure and function, the first subtopic is structure of model membrane. So this cell membrane is a double layered phospholipid molecule, has a double layered phospholipid molecule and some associated proteins and have some proteins. Okay. Then now due to this double layer phospholipid molecule, they are referred to as bilayer. Okay, they are referred to as bilayer. Now if this is a cell, if this is a structure of a cell, okay, having a nucleus, and here the cytoplasm is there where several organelles are present. This is a nucleus, and here there are several organelles are present inside the cytoplasm, okay, inside the cytoplasm, and this is a cell membrane. And this cell membrane is a double layered structure, as you can see here. Okay, so when we see a part of this membrane, what is the component of this double layered structure, we will observe now. So you can see here it's a double layered phospholipid molecule with a head and a tail structure, and some proteins are associated with it. Now, if you observe the structure of a cell membrane, how it looks like. So see, first of all, as I have shown you in the last slide, it has a head and it has a tail. Okay, the head region and the tail region. The head region is polar. So head region is polar and it is charged. This head region is made up of phosphate group. Okay, and the tail region is made up of fatty acid. Okay, head region is made up of phosphate group and the tail region is made up of fatty acid. These phosphate group are the charged molecules. So this head region are charged. Okay, and fatty acid tail. So remember, the head region is water soluble. And the tail region is not water soluble. Okay, the tail region is not water soluble. So the tail region is not water soluble. So the ones which are water soluble, it is said to be as hydrophilic. Head region is hydrophilic, and the one which is not water soluble, that is fatty acid, this is hydrophobic, which is not water soluble. Okay. Now remember one thing, this inside the cell there are lots of water, and outside the cell also there is water. So the cell membrane, so remember one thing, that outside the cell, what kind of environment is there? It is an aqueous environment. And inside the cell, what kind of environment is there? It is again aqueous environment. Okay. So we can say that this cell membrane is separating the external aqueous environment from the internal aqueous environment. Okay, the cell membrane is separating the external aqueous environment from the internal aqueous environment. Again, another thing to note down, that I told you that this lipid bilayer structure have the outer head or the polar charged head group, and which is hydrophobic in nature, as well as the fatty acid which is hydrophilic. hydrophobic in nature. Okay, the head is hydrophilic and the tail is hydrophobic. Okay, so the core part of the membrane is formed of what? The core part of the membrane, as you can see here, it is formed of lipid. Okay, now you can see here this hydrophilic region, and this is another hydrophilic region of the cell membrane. Okay, so if we see the cell structure of a cell membrane, so how it will be arranged? The head, this is the arrangement. So what we see, that one head is exposed to the external aqueous environment and another head is exposed to the internal aqueous environment, and the central core is formed by fatty acids. Since the central core is formed by fatty acid, then which kind of molecule can pass through the membrane? So the molecules which are lipid soluble, or the molecules which are very small, they are only able to pass through this lipid bilayer. That is from external to internal or from internal to external. So which molecules can pass easily? Which molecules can pass easily? The molecules which are either lipid soluble or the molecules which are very small. Okay, or the molecules which are very small. So what about the molecules which are charged? What about the molecules which are very big in nature? How they will cross the membrane? So the molecules which are charged, the molecules which are very large in size, they will cross the membrane by means of spatial transport proteins. Okay, we will discuss it later in detail. They will move by so this, say for example, this is a large molecule. How this large molecule will cross the membrane? It will cross the membrane by means of spatial transport proteins. Okay, this we will discuss later in detail. So let's discuss what we have studied till now. We have studied that the cell membrane is a bilayer composed of phospholipids and associated proteins. Phospholipids and associated proteins. Okay, how is the structure of the bilayer membrane looks like? They have the polar head, they have a head region and a tail region. Okay, the head region consists of phosphate group. They are charged, hydrophilic, they are water soluble or they are water loving. Okay, the tail region is composed of fatty acid chain. Okay, and they are hydrophobic. And this fatty acid region forms the central core of the membrane. Okay, then which molecules can cross the membrane? Okay, so which molecules can cross the membrane? So number one is the molecules which are lipid soluble. Okay, the molecules which are either lipid soluble or the molecules which have a very small molecular weight or very small size. Okay, they can cross the membrane very easily. But what about others? The molecules which are charged. Okay, the molecules which are very large, charged or polar, which are very large, they require spatial protein. They require transporters, or they require spatial arrangement for the molecules to move from the external to the internal or from the internal to the external region of the cell. Okay, up to here we have discussed till now. Next topic of discussion is asymmetry of lipid bilayer. Okay, so this lipid bilayer, this is asymmetric. What do you mean by that? So this lipid bilayer have two leaflets, outer leaflet and another is inner leaflet. Okay, and the composition of the outer leaflet and inner leaflet are different. Okay, they are not same. The first thing is they are asymmetric in the composition of lipid bilayer. Second is the proteins present on the outer leaflet is not similar with the proteins present in the inner leaflet. Okay, so the protein constituent of the outer leaflet and the inner leaflet is not same. Okay, the third is the carbohydrate moiety. The carbohydrate moieties which are attached to the outer leaflet, more significantly they are attached on the outer leaflet and it is either not present or they are different in the inner leaflet. Okay, now we need to discuss what are the lipid composition of the outer leaflet. So the composition of outer leaflet is the phospholipid which have choline group. Okay, so the phospholipid which have choline group forms the outer leaflet. So which phospholipid have the choline group? It is phosphatidyl choline and sphingomyelin So these two have a choline group and they are present in the outer leaflet. And what about the inner leaflet? The inner leaflet, the one which has primary amino group. They have primary amino group. So which phospholipids is having the primary amino group? They are phosphatidylserine and phosphatidylethanolamine. Phosphatidylethanolamine. Okay. Another important point which we need to remember that this phosphatidylserine, so what we saw that phosphatidylserine, this is present where? This is present in the inner leaflet. This is what we saw in the last slide. But in case of apoptosis, okay, in case of apoptosis, what happens? There is a change. In case of apoptosis, what we see that the cells which are undergoing apoptosis, they have this phosphatidylserine on the outer leaflet. Okay, so remember in case of apoptosis, this phosphatidylserine, in short I have written PS, it is present on the outer leaflet and in the normal case it is present on the inner leaflet. So let's revise what we have studied. We studied that the biological membrane, they are asymmetric in nature. They have two leaflets, the outer leaflet which is this one and the inner leaflet which is this one. And the composition of these two membrane is not same. It is asymmetrical in nature. It was seen in erythrocytes or in the RBC. It was seen that the outer leaflet have choline group. Okay, so the one which have choline group are phosphatidylcholine and sphingomyelin which are present on the outer leaflet, and the one which have primary amino group, that is phosphatidylserine and phosphatidylethanolamine, they are present in the inner leaflet. Okay, so the composition of protein in the outer leaflet and inner leaflet is different, means different types of proteins are present in the outer leaflet and inner leaflet. The protein composition is different and the carbohydrate moieties are mainly attached to the outer leaflet. So this is how they shows asymmetry in the nature of the lipid bilayer. And the another important point what we saw that Phosphatidylserine is present in the inner leaflet in the normal situation, but in the apoptosis, when the cells are undergoing apoptosis, this phosphatidylserine is present on the outer leaflet. Okay. So the next topic is cholesterol and the fluidity of the membrane. So cholesterol, this is a lipid soluble molecule. So this cholesterol, this is a lipid soluble molecule. I'm writing the characteristics of the cholesterol over here. It is a lipid soluble molecule. Okay, remember they have charged group towards the head side. Okay, they have charged group towards both ends. Okay, they have hydroxyl group towards both ends. Second thing, these cholesterol molecules, since they are intercalated between the two lipid molecules, then it decreases the fluidity of the membrane. So remember one thing, that these lipid molecules, they are dynamic in nature. Okay, these lipid molecules, I would like to write here about few points about membrane fluidity. So first thing is, this lipid molecules, they are dynamic in nature. That means they are able to move freely. They are able to move laterally with ease. Okay, the up down movement is like it is dependent on ATP. We will see that. So they are very easily, they move laterally. Okay, now these membranes, they are highly flexible in nature and they have the ability to change the shape. These membranes can change the shape because they have self-sealing property. Now, what is this self-sealing property? Since the lipid molecules are dynamic and they can laterally move very easily, they helps in self-sealing, and this self-sealing property is very useful in endocytosis, exocytosis, and membrane synthesis, that is membrane biogenesis. Clear guys? This self-sealing property of the lipid bilayer helps in endocytosis when they are engulfing anything, exocytosis when they have to release any content outside the cell, and the synthesis of the membrane. Okay, now what property of the membrane is helping them to move laterally? Because the bond interaction between the two molecule, okay, the bond interaction between the two molecules is non-covalent, because there is non-covalent interaction among the lipid bilayer. If it would have been covalent interaction, then there would not have been any lateral movement. So are you understanding guys? Now, since the covalent interaction is not there, these phospholipid molecules and the sterol molecules or the cholesterol molecules, so due to this non-covalent interaction, these phospholipid molecules and cholesterol molecules, they are able to move freely, or they have freedom to move. Okay, since these phospholipids and cholesterol, they can move, we can say that the lipid can exist in ordered state and disordered state. Okay. So let me summarize what we studied, that the biological membrane is dynamic in nature. Dynamic is due to because the lipid molecules, they can move laterally easily. Okay, the membrane can change the shape. They are self-sealing. They have the self-sealing property. That means they, after endocytosis or after exocytosis or during membrane biogenesis, they shows the self-sealing property. When there is any kind of pinching, then that is sealed automatically. Now, this movement is because the two lipid molecules have non-covalent interaction. This helps them to move. Now, because this phospholipid and cholesterol, they can move freely in the membrane, and because of this, the lipid can exist in ordered state and disordered state, which we will discuss in detail. Now, we will discuss in detail about the ordered phase and disordered state. Okay, so let's see. So if I say that this membrane fluidity, it depends both on number one, composition of the lipid bilayer, and number two, temperature. Okay, if we discuss about composition, so number one, composition of lipid bilayer. So what we see, that if the saturated fatty acid, if the fatty acid is saturated, so look here, composition. So for the ones which increases fluidity and the one which decreases fluidity. So the fatty acid which have long chain, okay, the fatty acid which have long chain and saturated fatty acid. So long chain and saturated fatty acid, they decreases the fluidity, because long chain and saturated structure, they pack well. Okay, they pack well and they increases the rigidity, which decreases the fluidity. The second is unsaturated fatty acid. So short chain and unsaturated fatty acid, this produces a kink in the structure. I'll show you how it looks. So this produces kink. So whenever a kink is produced in the structure, it interferes with the packing. So kink will interfere with packing. So the structure is not rigid at that time. This increases fluidity. Okay, this increases fluidity. So what we studied? We studied that the membrane is dynamic in nature. Its fluidity depends on the composition and temperature. Composition, when the fatty acid is short chain and unsaturated, it increases the fluidity. And when the fatty acid is long chain and saturated, it increases rigidity and decreases fluidity. Clear? Now, second point is cholesterol. Second point is cholesterol. The first point is about the saturation and unsaturation of the long chain and short chain. The second is about cholesterol. So whenever less amount of cholesterol increases fluidity, and when the cholesterol is present, it interferes with the free movement of the fatty acid chain. Okay, high level of cholesterol is present and decreases the fluidity. Okay, so when there is no cholesterol or very less cholesterol, then it increases the fluidity of the membrane. So look at this structure, what it shows. It shows that the flip-flop movement, which is the movement of the lipid from outer to inner and inner to outer. This is a rare type of movement. Okay, the lateral diffusion is a common type of movement. They also shows lateral diffusion and also rotation in their own place. Okay. Next you can see here in the unsaturated condition. In the unsaturated condition, what we see that there is a kink, that the tail is not straight. The tail is like the person is moving their legs here and there. Okay, so the person is now have started moving, that is they have introduced a kink. So whenever there is a kink, then they are still able to move. Okay, and whenever there is a kink, there is with cis double bonds. Okay, the cis double bond introduces kink in the structure, and this is a saturated one which is tightly packed. Okay, and this have saturated one have less fluidity, and the left one, which is unsaturated, they are loosely packed, as you can see, and have more fluidity. Next, the second point on which the membrane fluidity depends is the temperature. So what happens when the temperature is decreased or increased? So in the first case, if I make a chart, so at low temperature, what happens? So at low temperature, this bilayer exists in the semi-solid state. This bilayer exists in the semi-solid state, or better if I say semi-solid gel. Okay, now the motion of the cells or the motion of the lipid are restricted. Motion of lipids are restricted because they are gel-like structure right now. Okay, now what happens at the high temperature? What happens at high temperature? What we see? At high temperature, the fatty acids are in constant motion. They are in constant motion because of the rotation around CC bond. Okay, now the state at high temperature is a fluid state. So at high temperature, you all know that the fluidity will increase, and at the low temperature, the fluidity will decrease and the shape will be semi-solid gel at low temperature. Clear? So if I say about saturation and unsaturation, will you be able to tell me that at low temperature, will the cholesterol content be more or high? So at low temperature, cholesterol level will be high because it restricts motion. And what will about the fatty acid chain? So fatty acid chain will be long chain at low temperature, and the degree of saturation is more. Okay, but at high temperature, the fatty acid chain will be short and degree of unsaturation is more. I'm sorry guys, I wrote opposite. So here it will be Low temperature, where the structure is semi-solid gel, the molecules are tightly packed. So here long chain and saturated fatty acid, they pack well and increase the rigidity. Okay, this two factors increases rigidity. Okay, similarly short chain and unsaturation, they increase the fluidity. Another important point that these cholesterol molecules, they act as buffer. They act as buffer. So what do? They prevent low temperature from inhibiting fluidity and prevent high temperatures from increasing fluidity. Okay, so they work under both low temperature and high temperature. Normally under low temperature, what we see that the structure becomes rigid. Semi-solid gel is formed, the structure becomes rigid. So what is the role of the cholesterol as a buffer in under both of these conditions? We see that they prevent low temperature from inhibiting fluidity. What does that? At low temperature, what we see that it maintains the fluidity. Low temperature structure becomes, as I said, semi-solid in nature. They maintain the fluidity so that the structure does not solidify. Okay, and at high temperature, what it does to restrain, to prevent the structure to become excess fluid, what it does, it restrains the movement of phospholipids. It prevent the movement of phospholipid and reduces fluidity. So at high temperature they increases the fluidity and at low temperature they reduces the fluidity. In that way they act as a buffer for the biological membrane. So let's have a brief discussion about what we studied in this topic, that the cholesterol, they are the molecules which are intercalated between the lipid bilayer. Okay, they are intercalated between the lipid bilayer. They have the charged hydroxyl group which are present on the towards the outer side and the inner side. These cholesterol molecules, they are lipid soluble in nature. Clear? Now the membrane fluidity. So membranes are dynamic in nature, that the lipids are dynamic in nature, that the lateral movement of the lipid molecules are, or they happen very easily. Membranes, they can change the shape, they have the self-sealing property, and this self-sealing property helps the membrane to show endocytosis, exocytosis and membrane biogenesis. There is a non-covalent interaction among the lipid bilayer, as a result of which they can move freely. If it would have been the covalent interaction, there would not have been any movement. Now this lipid can exist in the ordered state, which is a rigid structure, and the disordered, which is the loose structure. Okay, next is what we studied, that the membrane fluidity depends on composition and temperature. The composition increases the either increases fluidity or decreases fluidity. When the lipid chain is short and unsaturated, then it increases the fluidity, and when it is long and saturated, it decreases the fluidity. So unsaturation produces a kink which interfere with the packing and increases the fluidity, and the saturated fatty acid chain, it increases the rigidity, because of it there is a tight packaging. Okay, now whenever cholesterol level is high, it can either decrease fluidity or increase fluidity. So I'm not including this point over here. Okay, I'm not including this point over here. because now you have understood that cholesterol acts as a buffer. Okay, now you have understood that the cholesterol acts as a buffer. It can either increase fluidity or it can decrease fluidity depending on the situation. So this point we will not include over here. Okay, next is what we studied, that the lateral diffusion is a diffusion process, that is one lipid molecule can move laterally either here or here, and they can do the rotation in their place, and the flip flop movement is very rare, that is movement of the lipid from the outer to the external leaflet or the inner to the outer leaflet. Another thing which we have studied is that the molecules in the unsaturated hydrocarbon chain, they have kink. Okay, this structure which you can see here, the leg is not straight, there is a kink, and you can see here that unsaturated hydrocarbon chain, there is a cis double bond. So remember, the cis double bond structure are more fluid. Okay, the cis double bond structure are more fluid. Now another one, the saturated one, they have the saturated hydrocarbon chains and they are tightly packed and have less fluidity. So that's it for the fluidity of the biological membrane. Lipid raft. So see here, what we have to know about lipid raft is, so the lipid domains, these biological membranes, they are not homogeneous mixture of lipid and protein. That means the lipid distribution is not random within a single leaflet. That means the sphingolipids, say for example the glycosphingolipid, for example cerebroside and ganglioside, for example glycosphingolipid, cerebroside and ganglioside, they contain long chain and saturated fatty acid. Here the fatty acids are tightly packed and they form clusters. Okay, they form clusters in the outer leaflet. So what we see, that this lipid distribution, it is not random in a single leaflet, rather there it is seen that there is a transient cluster formation in the outer leaflet. Okay, now this long saturated acyl group in the outer leaflet, it makes the structure more compact. What it does, it makes the structure more compact and stable. Stable means they can associate with long ring of cholesterol. Okay, they can associate with long ring of cholesterol. Now, so the structure will look somewhat like this, that the phospholipid molecule, the head group are tightly packed and the fatty acid chain are even, they are very close to each other. Okay, they are very close to each other. So as a result, what we see, that in some region there is a close packaging which is seen. Okay, there is a close packed structure which is seen, and associated with this close packed structure is a protein. Associated with this closed packed structure is a protein. Say for example, a receptor is associated with it. Okay, now what we see here, so we see that this outer leaflet, okay, this is more thicker and more ordered, we can say that, more thicker and more ordered. So when it is more ordered, it is less fluid than its neighboring microdomains. Okay, this region where the receptor is present, where there is a cluster formation, this region is more thicker than its neighboring phospholipids. Okay, now this whole structure is known as lipid raft. Okay, this whole structure in a phospholipid bilayer is known as lipid raft. So in a simple language, if I say what is lipid raft, so lipid raft are the receptors and some associated proteins. What proteins will be associated? Like for example, when we see any kind of signaling that the receptor binds with the ligand and intracellularly it will activate several other proteins. Okay, so some receptors, they are activating several intracellular proteins. Okay, so we can say that receptors plus their associated proteins, which is say for example the molecules or the protein molecules which regulate the intracellular signaling, which are rafting in the membrane. Okay, which are rafting in the membrane. Okay, not in the whole region of the membrane, but in a spatialized area of the lipid bilayer. In the spatialized area of the lipid bilayer. So what is written in this slide? That the biological membrane, they are not homogeneous mixture of lipid and protein. On the some side it is more thicker. Okay, within them are discrete domains in which certain lipids and proteins, they cluster together to form structural and functional units. For example, clustering together of cholesterol, sphingomyelin and glycosphingolipids in the outer leaflet with other inner leaflet give rise to lipid raft. So I hope the conception of lipid raft is clear. It is when the molecules are tightly packed, they have long chain. I write here, they have long chain and saturated fatty acid. They have long chain and saturated fatty acid and they are clustered in the outer leaflet, and we see that the receptors, this is a receptor, and their associated proteins, which is say for example intracellular proteins, they are rafting in the membrane in a spatialized area of the lipid bilayer. Okay, this structure is known as membrane or lipid raft. Now these lipid raft, they contain high number of GPI anchored proteins as well as palmitoyl anchored proteins. Okay, and high number of palmitoyl anchored proteins. So in this picture you can see that other phospholipid bilayer, here you can see that the neighboring microdomains, they are rich in phospholipids. So we can see here that this lipid raft, here is a cluster which is present, and this cluster we can see that this is less. This structure, what we see, it is slightly thicker than the neighboring phospholipids and they are more ordered. They are more ordered. Okay, they are more ordered, slightly thicker and less fluid. Since they are rigid, they are less fluid than their neighboring microdomains. So remember, the lipid raft have high number of GPI anchored proteins and high number of palmitoyl anchored proteins. Okay. So another thing we have to note down about lipid raft is that there are of two types of lipid raft. What are they? Number one is a planar lipid raft. Planar or non-cavular or glycolipid raft. So these planar raft are continuous with the plane of the plasma membrane. They are continuous with the plane of the plasma membrane. These planar lipid raft contains a protein named flotillin. Okay, and they are found in neurons. In which region of the neurons? Where caveoli is present. Where caveolar is absent. But one thing is similar between the two types of lipid raft is that the lipid composition is same. What is the lipid composition of lipid raft? I already discussed. It is cholesterol and sphingolipid. We will revise once again. The second is caveoli. So these caveoli, what is the characteristic? They are flask shaped invagination of the plasma membrane. Okay, and what proteins are present here? Caveolin proteins are present here. Where are they expressed? They are found in brain cells, microvessels of neuron system, microvessels of nervous system, in endothelial cells, astrocytes, oligodendrocytes, etc. Schwann cells, dorsal root ganglia, etc. So I'm not writing all. Okay, now one important point that both flotillin and caveolin both have the ability to recruit the signaling molecules into the lipid raft. So both flotillin and caveolin plays an important role in signal transduction. Lastly, what is the role of the lipid raft or the function of lipid raft? So these lipid rafts, they are involved in variety of cellular functions. Among them, one is membrane trafficking. So they are involved in membrane trafficking within the cells and they help in signal transduction. That is, they help in the transfer of the signals, extracellular signals, to the intracellular signals. Okay, transfer of extracellular into the intracellular region. Okay, so we can write here transfer of external signals into cellular response. Say for example, the receptor for insulin, or in short I write insulin receptor, and the receptor for growth factor, as well as certain GTP binding proteins. Certain GTP binding proteins and certain protein kinases. What do they all do? They are associated with the signaling. They are associated with the transmembrane signaling. They appear to be localized in the raft. They appear to be localized in the raft and in caveoli. Okay, so what we studied in this class is, let's revise once again. So what are lipid rafts? These lipid rafts, they are some kind of micro domains of cholesterol and sphingolipids. So they are micro domains of cholesterol and sphingolipids. Please remember these two. Okay, they sphingolipids and they form the lipid raft, and these lipid raft or these clusters, they are able to move laterally. These clusters, they can move laterally, or they shows lateral movement within the plasma membrane, and they are associated with some specific proteins. They are clustered in the specific region of the plasma membrane, and they are associated with some specific proteins. Clear? So here we discussed some receptors and their associated proteins which are rafting on the membrane in the spatialized area of the membrane is known as lipid raft, and they are the clusters. Okay, they are less fluid than their neighboring phospholipid bilayer. Okay, they are less fluid than their neighboring phospholipid bilayer. They are mainly made up of cholesterol and sphingolipids. Okay, next is, these lipid rafts have high number of GPI anchored proteins and high number of palmitoyl anchored proteins. We see that in the raft domain there is high number of more ordered structure, and the region is less fluid, more number of cholesterol is present. Okay, and the membrane is thicker. We see on the outer leaflet there is the presence of cerebrocyte and ganglioside, which makes them more stable because there are a large number of cholesterol molecules present there. Okay, next is what we studied, that there are two types of lipid raft. One is planar, which is non-caveolar, or glycolipid raft, which contains flotillin protein, and they are mainly found in neurons where caveoli is absent. Okay, another one is caveoli, which is flask shaped invagination of the plasma membrane, and the protein present here is caveolin. Whether it's planar or whether it's caveoli, both have same amount of cholesterol and sphingolipid. Okay, caveoli are found in brain, microvessels, nervous system, microvessels of nervous system, endothelial cell, astrocytes, dendrocytes, etc. And the function of lipid raft is, they help in membrane trafficking, they help in signal transduction, that is, they help the external signals to turn into cellular response. Some receptor like insulin receptor, growth factor receptor, GTP binding proteins, protein kinase, these are transmembrane signaling. They help in transmembrane signaling, and they appear to be localized in the raft and in caveoli. So that's it for lipid raft. Thank you. So today we will discuss about membrane proteins. So this membrane proteins play a very important role of the plasma membrane. Okay, these membrane proteins, they are distinguished into two types based on their arrangement. Okay, based on their arrangement, they are distinguished into two types. What they are? Number one is integral protein, and number two is peripheral protein. Okay, if I draw a structure of the lipid bilayer, so the first one, integral protein, these integral protein, which is this green colored one, this one is integral protein. Now this integral protein, they spans both the membrane. They spans both the outer and the inner plasma membrane. Okay, these integral proteins are also known as transmembrane proteins. Okay, and these integral proteins, they are attached with the lipid bilayer by hydrophobic interaction. Okay, they are attached by the lipid bilayer, or they are held with the lipid bilayer by strong hydrophobic interaction, due to which they are not easily extractable. Okay, so they are disrupted, or they are extractable by the detergents. They are extractable by detergents because they are held by strong hydrophobic interaction with the lipid bilayer. Now coming to the next one, peripheral protein, which is the blue colored one. This peripheral protein, they can either be present on the extracellular side or in the intracellular side. Okay, they are present in the peripheral region. That's why the name is peripheral proteins. And these peripheral proteins are either present in the extracellular side or the intracellular side. And they are very loosely arranged with the lipid bilayer. Okay, they are held to the lipid bilayer by non-covalent interaction. And this non-covalent interaction is a very weak interaction. So by just changing the pH, we can extract the peripheral protein. Okay, so it is extractable by changing the pH only. Okay, since there is a loose bond between the peripheral protein and the lipid bilayer, it can be extractable by changing the pH. Okay. Now the next part is composition of the cell membrane. So how much protein, how much lipid and how much carbohydrate is present in the cell membrane. Okay, so there is 50% of the protein. The composition of the cell membrane is 50% of the cell membrane is occupied by protein, out of which the lipid is 48% and carbohydrate is just 3% in the cell membrane. So the cell membrane is composed of 50% of proteins, around 47% of lipid and 3% of carbohydrates. Okay?
By mass. Membrane proteins come in two flavors, integral or intrinsic, and peripheral or extrinsic. Integral membrane proteins are a permanent part of the membrane, while peripheral proteins are only transiently associated with either the membrane or integral proteins, where these associations are hydrophobic, electrostatic, or other non-covalent interactions. There are several different kinds of integral proteins. Integral monotopic proteins are attached to only one of the two leaflets of phospholipids making up the membrane, and they don't span across it. There are also transmembrane proteins and lipid-anchored proteins. Transmembrane proteins are those that span the lipid bilayer, and can be bitopic, spanning across the membrane once, or polytopic, spanning across it more than once. Lipid-anchored proteins are those which are covalently attached to lipids embedded in the lipid bilayer. For example, GPI, or glycosyl phosphatidylinositol, is a glycolipid that gets attached to a protein C terminus during post-translational modification. It acts as an anchor for proteins to the outer leaflet of the plasma membrane. Both integral and peripheral proteins can be post-translationally modified. There can be addition of fatty acids, diacylglycerol, prenyl chains, or GPI. Recall that cellular membranes are made up of a phospholipid bilayer, which consists of two leaflets of phospholipids. These phospholipids have polar heads which are hydrophilic, or water-loving, and nonpolar fatty acyl tails that are hydrophobic, or water-hating. Polar substances like to interact with other polar substances, and nonpolar substances hang out with other nonpolar substances. This really attests to the power of hydrogen bonding. Water molecules want to interact so badly with their polar buddies that anything nonpolar getting in the way of their hydrogen bonding results in decreased entropy. The result is what's called the hydrophobic effect. This is why phospholipids in water will spontaneously form lipid bilayers. These bilayers minimize contact between polar and nonpolar molecules, maximize hydrogen bonding, and maximize entropy. This is also why transmembrane proteins are amphipathic, which means that they have regions which are hydrophilic and regions which are hydrophobic. The hydrophilic regions are exposed to water on either side of the membrane, while the hydrophobic bits are happily interacting with the hydrophobic tails of lipid molecules in the interior of the bilayer. As a result, transmembrane proteins are stuck permanently in the cell membrane and are very hard to isolate. To get them out, you need to add detergent, which is amphipathic and will disrupt the lipid bilayer. There are two basic types of transmembrane proteins, alpha helical proteins and beta barrel proteins. Note that while helix bundle proteins are found in all types of biological membranes, beta barrel proteins are only found in the outer membranes of gram-negative bacteria, mitochondria, and chloroplasts, evidence that contributes to the endosymbiotic theory, in which eukaryotic cells acquired these organelles through the ingestion of prokaryotes. Transmembrane protein structure can be predicted using a hydropathy plot, which has hydrophobicity index on the y-axis and amino acid number on the x-axis. The amino acids making up a protein are localized according to polarity within its final structure, in such a way that the polar amino acids face the outside aqueous solutions, and the nonpolar amino acids are adjacent to the lipid bilayer. Transmembrane proteins can be classified by topology, which is based on the position of N and C termini, as well as start transfer and stop transfer sequences. For example, type 1 is a single transmembrane pass with the N-terminus on the extracellular side of the membrane. Type 2 is also a single transmembrane pass, but the N-terminus is on the cytosolic side of the membrane. How these different topologies come about will be the topic of another video. Often, transmembrane proteins function as gateways, allowing specific substances to pass across the membrane. They can undergo conformational changes as they do this. They might participate in facilitated or active transport. Facilitated transport is spontaneous passive transport of substances via transmembrane proteins. Active transport, however, requires energy. Active transport may be necessary, for instance, if a substance is being carried across the membrane against its chemical or electrical gradient. As a final note, in animal cells, most transmembrane proteins are glycosylated. These sugar residues are always present on the non-cytosolic leaflet of the membrane. As a result, the cell surface is covered in carbohydrates, which form what's called the cell coat.